Halomonas Maura, Biological Solution of Sand Laws by Jason Lee. I will go through my motive, what coastal erosion is, what mudflats are, and lastly the goal of this experiment. I visited my friend's house in Cape Cod last Thanksgiving and I was astonished by the coastal erosion in the area. Most of the shores were gone and some houses were in danger from the water. After I came back home, I did research on the solutions for coastal erosion, but most of them were physical and short-term solutions using breakwaters. Coastal erosion is a phenomenon of erosion taking place near the land of adjoining the sea, whereby sand is transported by such causes as weather and storms, and which causes changes in the coastal topography, and even causes topographical changes in the flow of waves to change in the coastal area. Mudflat is characterized by the fact that it is not easily washed away due to viscosity even when the water continues to move. The viscosity of the mudflat is formed by the action of various microorganisms present in it, and thinking that it has a particularly high bacterial action, I decided to use it to separate the mudflat bacteria and increase the sand's viscosity. In this study, I have attempted to delay the coastal erosion by increasing the sand's viscosity and decreasing the sand's movement, since coastal erosion is caused by the sand's movement by wind and wave. I decided to use mudflat bacteria as a means to increase the sand's viscosity. The experiment proceeded as follows. Now I will explain the results. In order to isolate tidal flat bacteria, bacterium colony was identified by cultivating marine agar in Gangwa Island. The bacteria were classified into 13 species using colony form, margin, elevation, and color. In order to use tidal flora in the sand, it was confirmed that the bacteria could grow in the sand. The sediment of the sand was mixed with the marine culture medium, and the growth of the bacteria was confirmed. Comparing the growth of the bacteria, the number of bacteria in the MB containing sand components increased relative to that of NB, and the bacteria showing growth were A, B, D, E, and L. I examined whether the sand's cohesion increased by the bacteria by comparing the turbidity of the water after inoculating the mudflat bacteria in the sand and flowing the water. Bacteria A, B, E, J, K, L, and M, whose degree was lower than the control which did not treat bacteria, each showed values of 1.164, 1.251, 1.184, 0.971, 0.505, 1.222, and 0.623 respectively. Therefore, bacteria A, B, E, J, K, L, and M were likely to play an important role in increasing the sand's cohesion in the sand environment. Because various bacteria act in the natural environment, various bacteria combinations were made and experimented. Combined two species of bacteria using bacteria A, B, E, J, K, L, and M, which strengthened the sand's cohesive force and verified the sand's cohesion through turbidity of crumblings. Therefore, when using the mudflat bacteria in the sand, it would be possible to increase the cohesive force by using two species of bacteria together, not only with one species, and if the combination of bacteria which showed stronger cohesion when two species of bacteria were used together, can be used together without interfering the growth of each other. I verified which bacteria A, B, C, J, K, and M can live in the nitrogen environment in order to investigate the characteristics of bacteria which increase the cohesion of sand. Bacteria E grew in a large quantity, and bacteria J did not grow at all. A large number of colonies were observed in both bacteria K and M. Therefore, it was found that bacteria E, A, K, and M are strong in the nitrogen environment. Exopolysaccharide, EPS, is a substance which synthesizes bacteria and secretes them to the outside and is one of the substances which contribute to increasing the viscosity as a polymer substance. Then the bacteria formed EPS. I thought they could contain sugar components or particles would be observed by polysaccharides. The gene was analyzed by identifying the bacteria that increased the viscosity of the sand. 16S ribosomal RNAs were analyzed and verified by using the BLAST. The mudflat bacteria A, Halomonas Maura, which is a salt-loving bacteria and is one of the most common bacteria in the mudflat environment, and has excellent metabolism, further to being known to form EPS. It also has the ability to fix nitrogen under a wide range of salinity. Mudflat bacteria B is Bacillus metagrium, which inhibits at temperatures of 3 Celsius to 65 Celsius and is used as an important industrial organism and is used for biological control of plant diseases. In conclusion, I isolated 13 species of mudflat bacteria. A, B, D, E, and L increase in sand environment. Bacteria A, B, E, J, K, L, and M increase sand cohesiveness. And lastly, exopolysaccharide increases viscosity and cohesion. So, I would like to fuse them with artificial intelligence, create a device which can detect them by AI in the sea, and also calculate how much of bacteria are needed and supply where they fall short. People can just check the big data on their computer, and the robots can naturally inoculate the bacteria in the spray, formed to increase the viscosity of the sand and maintain a cohesion. Thank you.